درود مجدد به آدینس محترم که امروز به دورهمی علمی ما در کلاب هاوس پیوستین. سلام میکنم به دوستان مادریتور، همچنین خوش آمد میگم به دکتر کیوان که قبول کردن امروز در این کلاب در خدمتشون باشیم و از دانش و تخصصشون یاد بگیریم. این اتاق 35مین دورهمی علمی ما هست در کلاب هاوس. از چندین ماه گذشته سعیمون بر این بوده که از متخصصین و محققین هر هفته دعوت کنیم در زمینه‌های مختلف که به کلاب ایرانیان هاروارد بیان و با بحث‌هایی که انجام میشه از دانش و تخصصشون یاد بگیریم. امروز هم مهمون ویژه‌ای داریم. ایشون از محققین و متخصصین برجسته در زمینه متابولیسم سرطان، تشخیص و درمان سرطان با استفاده از روش‌های غیر تهاجمی و تغییرات متابولیک، همچنین Hyperpolarized MRI که نوعی عکسبرداری هست که برامون توضیح خواهد داد. امروز دکتر کیوان کشاری، زیست شیمیدان و مهندس زیستی و همچنین عضو هیئت علمی در مرکز سرطان مموریال اسلون کترینگ امروز در کنار ما و شما عزیزان هستند که به سوالات ما مرتبط با تحقیقاتشون، زندگی و حرفشون پاسخ بدن که مطمئنم بحث جالبی خواهد شد، چرا که خیلی از ماها آرزوی پیدا کردن راهی مطمئن برای تشخیص زودهنگام و درمان و در نهایت معالجه سرطان رو داریم. دکتر کشاری هم سال‌هاست در این زمینه فعالیت دارند، همچنین در یکی از قدیمی‌ترین و مجهزترین مراکز تحقیق سرطان در دنیا فعالیت علمی دارند. ایشون سرپرست آزمایشگاه کشاری در آزمایشگاه‌های تحقیقاتی بیمارستان مموریال هستند و عنوان محقق برتر رو از آکادمی رادیولوژی و تحقیقات تصویربرداری زیست پزشکی در سال 2020 دریافت کردند. این جایزه به افرادی داده میشه که بیشترین تأثیر رو در این رشته در بحث تحقیقات آکادمیک دارن و خب به واسطه کارهای تحقیقاتی ارزشمندی که انجام دادن جوایز متعددی رو هم دریافت کردن. این گفتگو مطابق گفتگوهای هفته‌های قبل ضبط میشه و شما میتونید در کانال یوتیوب کلاب و همچنین سایر سوشال میدیاهای کلاب در واقع دنبال کنید. اگر به سایت کلاب برین میتونید از اونجا به تمام در واقع کانال‌های اجتماعی کلاب دسترسی داشته باشید. من خیلی کوتاه فرمت اتاق رو خدمت شما عرض میکنم و بعد در واقع اتاق رو شروع میکنیم. ابتدا دوستان مادریتور سمانه جان و محمد جان خودشون رو معرفی میکنن خیلی کوتاه تا فرصتی باشه دیگر دوستان به جمع اضافه شن و بعد دکتر کیوان خودشون رو معرفی میکنن و با سوالی از ایشون جلسه پرسش پاسخ رو شروع میکنیم و دوستانی هم که در اتاق هستن میتونن در واقع سوالات خودشون رو در اسلایدو وارد کنن. همین در واقع اکانت اینفو که در روی استیج هست یه لینکی روی پروفایل پیکچر داره bit.ly/dr_keyvan. سوالاتتون رو اگر تو این اسلایدو وارد کنین یا اگر براتون امکانش نیست میتونید تو بک چنل بفرستین و دوستان در حین بحث از کیوان عزیز میپرسن. مدت زمان برنامه 2 ساعت هست. اگر که سوالات بیشتری باشه و ایشون وقت و زمان انرژی بیشتری داشته باشن خوشحال میشیم که در کنارمون باشن و به سوالات شما پاسخ بدن و در انتها هم میریم سراغ حرفهای پایانی ایشون و دوستان مادریتور. میریم سراغ معرفی مادریتورها. از خودم شروع میکنم. در دانشگاه هاروارد در دپارتمان محاسبات تحقیقاتی سرپرست تیم مهندسی نرم افزار پژوهشی هستم و خیلی خوشحالم که امروز در جمع شما عزیزان.
مرسی محمود جان. من هم سلام میکنم به شما و همه دوستان حاضر در این اتاق و یک خوش آمد ویژه میگم به دکتر کیوان عزیز. من سمانه راد هستم، استادیار دانشکده مهندسی در مؤسسه فناوری نیوجرسی و هدف اصلی گروه تحقیقاتی من توسعه مدل‌سازی چند مقیاسی و روش‌های شبیه‌سازی جدید برای مطالعه مکانیک و فیزیک سافت متر یا ماده نرم، انتقال ذرات سلولار و جریان‌های بیولوژیکی است و در حال حاضر هم تمرکز گروه من روی طراحی و بهینه‌سازی نانو کریرها یا نانو ذرات حامل دارو و یا آنتیبادی و مطالعه اینتراکشنشون با سلول‌های درگیر هستش. محمد جان بفرمایید.
سلام، وقت همگی بخیر و خوش آمد دارم خدمت دکتر کشاری عزیز. محمد زاری هستم، توی دپارتمان مدیسین دانشگاه هاروارد مدیکال اسکول و همینطور در مرکز جان بیکر در دانشکده سلامت دانشگاه هاروارد مشغول فعالیت توزیع تولید دارو هستم. محمود جان برمیگردیم به شما.
مرسی سمانه جان و محمد جان که هر هفته میاین به بحثا کمک میکنید. احسان رو هم میتونم که تا دیر وقت دیروز در دانشگاه بود و حالا اگر امیدوارم که در ادامه بحث به جمع بپیونده. میریم سراغ دکتر کیوان عزیز که خودشون رو یکم جزئی‌تر برای دوستان معرفی کنن در واقع از اون سالهایی که در واقع مشغول به لیسانس دوره لیسانس رو شروع کردن، اون مسیر تحصیلی و شغلی که داشتن که در واقع کمک کنه به بحث و بعد بریم سراغ در واقع تحقیقاتیشون. آیکون میکروفون هم پایین سمت راست هست.
بله مرسی. so اگه لطف کنید منم اینو در انگلیسی میگم که یه ذره برای من آسان تره. حالا اگه یکی میخواد یه چیزی بپرسه تو فارسی منم میتونم جواب بدم، ولی یه ذره تندتر تو انگلیسی حرف I wanted to say thank you for inviting me. I actually wasn't aware of Clubhouse until you emailed me, and now I'm learning about it, which I think is a really interesting thing. So thank you for the invitation and for the opportunity to talk to you, and hopefully I can help in any way I can if there are things that you'd like to know. So in terms of introducing myself, I did my undergraduate at Berkeley in I was a dual major in biochemistry and applied mathematics, and actually it's kind of just as a funny aside, I don't know if I'm sure many of you saw that Google changed its search engine picture the other day to Malala Yousafzai, and he was a professor at Berkeley when I think just kind of going emeritus when I was a student, and so it's kind of nice to see that recognition. After undergrad, I did a stint in research and then started a joint PhD between a lab at the University of North Carolina to study bioengineering and a lab at UCSF to study imaging. I ended up finishing my PhD at UCSF, and then I did a postdoc there. And in my fellowship, I did more imaging than I had done engineering. I mean, you could consider imaging kind of a subsidiary of engineering. And I left UCSF in 2013, about the early 2013, and I started my lab at Kettrring at about that time. And the goal of my lab right now is to really try to understand biochemical mechanisms in cancer, to use them to develop imaging modalities, imaging tracer strategies, and also therapeutic strategies to try to address cancer. And so we explore a lot of these methodologies kind of from an engineering side, physics side, as well as fundamental biochemistry, chemical biology side. So my lab is kind of split in half in that respect. Many of the students that join my lab so we have a joint program with Cornell, and so I'm also a professor at Cornell, and so a lot of our students come from there, and so we have a number of students that come in that are either biochemistry students, and I'm in the biochemistry program, but also biophysics and chemical biology and pharmacology, and I think those students kind of fill in the long-term goals of the lab that many of the postdocs also work on, and so that's generally what my lab is interested in, and kind of the directions that we've been going in. And I'm happy to talk more about any of these aspects that you would like to learn more about.
خیلی ممنون. بسیار عالی. آره مطمئنا خب این سالهایی که زیادی که اینور بودین انگلیسی راحت‌تر هستین. حالا ما هم سعی می‌کنیم که میکس فارسی و انگلیسی هر جا که سوال‌ها سعی می‌کنیم اون اصطلاحات رو حداقل اصطلاحات علمی رو به انگلیسی بگیم، هر جا که براتون راحت‌تر بود بگید که حالا سوال رو به انگلیسی بگیم. خوشبختانه ما دوستان هر هفته لیست سوالاتی که توی همون اجندا آماده می‌کنن تماما به انگلیسی هست و حالا کار ما راحت‌تر می‌کنه. ممنون میشم که اگر حالا همین ریسرچ که در واقع ریسرچ رو یکم به زبان ساده‌تر توضیح بدین که در واقع چی شد که به این موضوع علاقه پیدا کردین، دقیقاً دارید تو این فیلد چیکار میکنین و خب مطمئنم که به جریان بحث تو اتاق کمک میکنه.
Simply, or maybe the most simple kind of description of this is that we are interested in how nutrients flow through your body, how can we trace them, and what do they turn into in time. So this is a fundamental implication for almost every pathology that we are interested in, and also provides a mechanism to address them. And so on the kind of most simple level, we try to trace the fate of everything that they sell at GNC. Where do these sugars go? What do these sugars turn into in real time? And can we develop strategies to visualize it? So I think right now, the methodologies to understand dynamics in vivo are actually very limited. We can't see things go from, one thing to another, get converted through a reaction in vivo. It's hard to quantify those. And we're interested in trying to understand that biochemistry. And so it takes a lot of engineering and physics to develop ways to see it, but it also takes a lot of biology, biochemistry to understand what you're seeing. And I think that's the basis for what we're kind of driving this field. So a lot of the work that, probably 50% of what we publish is mostly in journals that are kind of cancer metabolism or, whatever nature cell kind of focused. And the reason is because we're trying to use these tools to understand these basic kind of fundamental questions about how fast something happens and where does it happen. And if you block it, how does something respond? And I think it's surprising, but these things we've been talking about for 100 years, we actually don't know the answers to those questions. And so I think if you're in a space where you're trying to ask kind of basic questions that are hard, and try to answer questions that apply to a lot of things, then they become kind of higher impact maybe. But most of the recent things that we've studied are related to sugar metabolism, like how do certain cancers use sugars in different ways and what do they do with them? How do certain vitamins, like we were interested in reducing an oxidation or what's called redox, and how do reactive oxygen species change metabolism? How can you understand that chemistry? And so we ask chemical questions or kind of biochemistry questions, but we try to answer them with biophysics. So it's kind of a combination thing.
مرسی از شما. پس به نوعی که اون در واقع سلول های سرطانی، متابولیسم سلول های سرطانی و نوعی که حالا اون در واقع شوگر رو استفاده میکنن، تفاوتی که داره با سلول های دیگه، شما سعی میکنید که اونا رو ترک کنید. حالا این ترک کردنتون به چه صورت هست؟ میشه یکم بیشتر توضیح در مورد اون روش ایمیجینگ؟
So we typically use magnetic resonance as our basis for detecting things, or MRI. And what we kind of add to that is a new field of MRI that's called hyperpolarized MRI. And the idea there is that magnetic resonance is fundamentally sensitivity limited in that, relative to other techniques, PET, SPECT, whatever radioactive approaches, which are very sensitive, meaning like you don't need very much to detect something radioactive. MRI is not in that space. It's actually very insensitive, though we do a lot with it. To overcome that insensitivity, because we want to see these small concentration, micromolar, nanomolar things, these metabolites, and reactions happen fast, but they do not make that much stuff. So the question is, how do you overcome that insensitivity? The way we do it is by changing nuclear spin states of molecules outside of a magnet. And so that's called hyperpolarization, because there's an inherent Boltzmann polarization for nuclear spins at a given field. And so what we do is we change that polarization state for a given molecule. So we develop the physical chemistry, use some kind of, low temperature physics to change the polarization state of a molecule. And then once it's polarized, we infuse it into a living system. So now we introduce this molecule to a living, either whatever, mouse, person, cells, whatever it is. And then use actually fast MRI to image what that molecule is doing, where it goes, and also use spectroscopic imaging to resolve what it turns into. And so we can do that by using the fundamental properties of NMR. And so that allows us a non-radioactive strategy where we can image things using conventional MRIs, but we can enhance them by using these hyperpolarized molecules. And so that's why the lab is kind of interested in a lot of chemistry, because we develop chemistry to hyperpolarize things and then use that chemistry to allow the physics that we use in the lab. So that would be like the MRI physics. And that's how these two fields come together. Much of the work that we have done is in trying to understand biochemistry of the tools, but also translate it to humans, because being able to image people and image dynamics spatially resolved in a person provides context for trying to understand disease. If we couldn't do it in people, then it would kind of just be a tool for scientists to use in the lab. So it's kind of not just a tool, but a translational tool.
بسیار عالی، مرسی از شما. حالا در مقایسه با خود MRI، این Hyperpolarized MRI چقدر در واقع قدرت تشخیصش بیشتر هستش در اگه بخوایم مقایسه کوانتیتیتیو بکنیم؟
So it would be so for carbon, which is what we normally do. So typically MRIs, you use protons of water, which are high concentration, ubiquitous. So water is 55 molar. There are 2 protons, so it's 110 molar. We inject substrates for chemical reactions in the millimolar concentration, which get diluted to micromolar, but we see them with the sensitivity of water. So they are enhanced 10 to 100,000 times stronger than what they should be. And there's no background signal because we only see what we inject, because nothing else is a strong enough signal to register. So we can do real-time MRI without signal averaging because the signal is so strong. But the problem we have is that it doesn't last forever. So the signal it's decaying as we're imaging it. So it has some properties similar to what you would expect from a radioactive nucleus, except it's not radioactive. It's just decaying as a function of its spin state going back to equilibrium. And so we have to image it quickly, but we have a very, very strong signal, like on the order of 10 to the 5th, stronger than kind of conventional MRI.
خیلی ممنون. برای این در واقع روشی که استفاده میکنید Hyperpolarized MRI در واقع روش تصویر برداری هست که طبق گفته شما توانایی بیشتری از MRI داره برای در واقع این که شما فیگر اوت کنید که چه تغییرات در واقع شیمیایی اونجا اتفاق میفته. در چه مرحله هستید؟ آیا در مرحله پریکلینیکال هستید؟ در چه فازی از تحقیقات هستید در این؟
So we're in both preclinical and clinical imaging. So we are doing this already in humans, where we make these molecules and inject them into people. But we're also developing new things preclinically for the purposes of eventual translation, or just to understand how things work. I mean, you could imagine that there is a fundamental mechanism which you cannot figure out using other methods, that something like this would be able to answer that question. Or maybe the other methods are very hard, challenge to execute, and maybe this is something that we can do that can answer a question about biology or chemistry or physics that maybe we couldn't use another tool to address. And so I think there's kind of this dichotomy. I mean, it's very similar to MRI in general. I mean, you could think of this as, turbo MRI. The MRI can answer lots of questions in material science and in fundamental physics, even though we also have this duality that we use it for imaging people. And I think a lot of the imaging of people is what finances a lot of the basic science that people do with MRI. So hyperpolarized MRI is very similar in that respect, that it also benefits from this kind of clinical aspect.
مرسی، خیلی ممنون کیوان جان. من برام جالب بود که راجع به اپلیکیشنش بدونم، یعنی در حال حاضر این نتایجی که شما میگید حتی تو مرحله انسانی هم رسیده، روی چه بیماری های آزمایش شده و در واقع کاربرد کنونیش چی هستش؟
So predominantly cancer. We're in the clinical setting. People have studied mostly cancer, but a lot of work in cardiac disease, as well as just brain imaging. And people are very interested in neurology and neurodegenerative, diseases. Preclinically, this space is very large. Basically anything, any disease pathology that you could model in a mouse or a rat or something has been placed in a magnet and studied with some of these probes that we've developed over the years. And I think people have learned quite a lot about it. So, but translation is very open. But right now the cancer in some ways is easy. That's why we did it first, because it's nodular, localized, can be perfused. It's a big problem. There's a lot of reasons why studying cancer with this approach is a good idea. And maybe the easiest way to get it through the FDA, which is why I think we're kind of pushing in that direction.
مرسی، خیلی ممنون. خیلی دوست داشتیم که یه مقدار راجع به این مؤسسه MSK بدونیم، اینکه اولا چه فیلدایی توش کار میشه. من یادمه که سعی میکردم چند سال پیش با یکی از اساتید اونجا تماس بگیرم که روی بحث بایوفیلم مدییتد اینفکشن کار میکردم. دوست دارم بدونم تو چه فیلدایی کار میکنن و اینکه هایر کردن دانشجو به چه صورت هستش، اصلا چه لول پروگرامو شروع میشه، و مطمئنم تو این اتاق کسایی هستن که دوست دارن راجع به این هم بیشتر بدونم.
Of course. So Sloan Kettering. So my lab is in the Sloan Kettering Institute, which is the research arm of Memmoorial. It's a kind of a two-part place. There is a hospital called Memmoorial Hospital, and there's an institute called Sloan Kettering Institute, which with departments. At the postdoc level, it is there are labs in all of these different departments and people studying all kinds of things. Most of the departments are biologically focused, so biochemistry, chemical biology, cell biology, cancer biology. The most kind of translational or kind of technical department would be pharmacology, which is the program that I'm in. And so at the postdoc level, there are just labs strewn across this, and all with a focus in cancer. So I think that's the unique thing about Sloan Kettering. We are not a big place. We're a standalone private cancer hospital or cancer institute. We have about 150 labs. We're, I think, the oldest standalone. I think we're the first standalone cancer center in the world. Well funded. And we typically compete with the Ivy League schools, both for faculty and for students. On the graduate side, we have a very small graduate program internally at Sloan Kettering that gives you a PhD in cancer biology. But we have, I think, 9 joint graduate programs with Cornell across all of the biological sciences. So we offer a PhD in biochemistry and molecular biology, pharmacology, immunology, genetics, structural biology, biophysics, chemical biology, and we also have a tri-institutional MD PhD program where we offer students get their MD from Cornell and they get their MD they do their medical training at Cornell and then they do all of their PhD training at either us or Rockefeller University. Which is, and these 3 universities are just across the street from each other, which is, I think, very powerful. And so most of the students tend to be interested in these basic sciences with a kind of a cancer focus. I would encourage anyone who has any interest in cancer, regardless of their background, if you're an engineer, I was a trained engineer, or a physicist or a chemist, don't be afraid to apply to one of our programs. We would love to take technically minded, undergrads and make them, applied in the cancer fields. I think that's something we're very excited about. And so I would encourage you to take a look at these websites. Actually, right now, I don't know if the applications have closed for graduate students, but reach out, email any of the faculty. They would love to talk to you. And don't let what the name of the place is fool you. It's a very strong kind of university institute, And so we have a big push for education.
خیلی عالی. مرسی از معرفی مرکز تحقیقات سرطانتون. منم توی چندین تا وبسایت میخوندم از اینکه خب یکی از قدیمی ترین مراکز هست، بیش از 130 سال اگه اشتباه نکنم، و خب خیلی این کلابوریشن که شما با دانشگاه‌های اطراف دارید، دانشگاه کرنل دارید، میتونه خیلی کمک کنه. خیلی خوش آمد میگیم به احسان عزیز و هادی عزیز. میدونم که هادی جان میتونه صحبت کنه، حالا یه اصلاحاتی هم برای کیوان جان بشه. هادی جان ممنون میشم که خودتون رو معرفی کنید و سوالی دارین بفرمایید.
سلام، خوش آمد میگم به کیوان عزیز. من هادی هستم. من تحصیلاتم رو توی رشته الکترونیک از ابتدا تا پی اچ دی انجام دادم. پی اچ دیم رو توی دانشگاه کرنل بودم و بعداً اومدم توی گوگل. الان 6 ساله که تکلیت هستم توی بخش هاردور کروم او اس. سوالی که به ذهنم رسید از همین صحبت‌های کیوان عزیز این بود که شما فرمودید که مهندسین انجینیرها هم میتونن اپلای کنن توی مرکز شما. از دید یه مهندسی که کامپیوتر ساینس یا الکتریکال انجینیر خونده، چه اپلیکیشن هایی هست، چه کارهایی هست که اون مهندسان میتونن توی مرکز شما انجام بدن؟
Sure. So both, I would say, biochemical engineering as well as electrical CS would I think find a great home in our labs. And from the setting of electrical engineering CS, it would be mostly in image analytics, image processing, image acquisition, development of detectors. We do a lot of kind of building of things, I would say, lab on a chip type stuff, and also in vivo imaging types of applications. So I would, for the students that, at the level of postdoc, I think my lab, for example, always has at least one or two kind of electrical engineering, people who did their EE at Stanford or Berkeley or wherever, and they're postdocing in the lab. And their projects have ranged from kind of all of those things. But I think at the graduate, if you're an undergraduate in EE, it gives you an opportunity to really build your understanding of biochemistry and chemistry to use your EE in that state. And, when I was a graduate student, the joint graduate program at UCSF Berkeley actually originally came from EE. The bioengineering program was actually a major in EE. So when I was an undergrad, there was no bioengineering major. It was made afterwards, but it grew out of kind of EE. So I think a lot of the work that we do is kind of bio-E with an EE spin in that respect. So, there's built-in signal processing and things like that.
بسیار عالی. مرسی از هادی عزیز از سوالتون و کیوان جان از پاسخ کامل. زمینه‌هایی که شما کار میکنید برای در واقع تشخیص زود هنگام سرطان خب خیلی پتانسیل در واقع ترنسلیشنال ریسرچ داره. بتونید شما حالا اینو کامرشالایز کنید، تبدیل به پروداکت کنید. آیا شما هم در همین بحث آنترپرنورشیپ فعال هستید؟ دنبال در واقع استارتاپی کار کردین یا حالا اگر در این مورد نکته‌ای دارین بفرمایید.
Yeah, so this is a great question. I think when I started the lab, I was more worried about making the lab work. I think when you're an assistant professor, there's no class on how to run a lab. It's, you go from one side of the table where you would show up with your lab notebook or whatever and, discuss with your advisor and, I don't know, have disagreements, and you would leave and go do experiments and come back and you would work on your things. And I think when you change seats, you don't realize how many things change. It's kind of like growing up. And so I think when I started the lab, I was most interested in trying to figure that out. And in the last 5 years, we're interested in this entrepreneurial aspect of the lab. And so I sit on boards of companies, but also most recently we're thinking about starting another company to help accelerate some of the things that we're working on in the lab. to get them out there. And I think the field is now getting there, both from the standpoint of molecular imaging and also from the standpoint of therapeutics that are related to molecular imaging strategies. And so we've been starting to do a lot more of that. So I am personally very interested in that, and I've started to get interested in getting my postdocs to want to do that as well. And so some of them have actually gotten very excited about starting their own companies, which I'm also very supportive of. And so I think it's a great aspect of research. Is another direction. Not everybody that gets a PhD wants to be a professor. A lot of ways it's very challenging right now to do that transition. The requirements, especially if you want to do it when you're young, if you want to go down that road as an academic at an early, like kind of right out of postdoc, requires it's much harder now. And so I think, and a lot of people are not interested in what's required to do those things. And so I think that this entrepreneurial angle is a massive positive for a lot of people who have PhDs and now they're changed their mind. Maybe they don't want to be an academic.
بله خیلی هم عالی، مرسی از توضیحاتتون. من یه سوالی راجع به همین بحث HPMRI داشتم. خب میدونید یه نکته مهم برای در واقع اون کلینیکال اداپشنش، اون پذیرش بالینی این HPMRI همون بحث هزینه هستش. میخواستم یه مقایسه‌ای داشته باشیم راجع به هزینه این بحث همین HPMRI در مقایسه با پت مثلا چقدر متفاوته، چون توی یه مقاله‌ای میخوندم که تعداد این دستگاه تکنولوژی HPMRI مثلا هولوش 50 تا هستش تو کل دنیا. حالا نمیدونم عددش درست هست یا نه، ولی معنیش یعنی برای من معنیش این بود که شاید این پرهزینه هستش و فقط به مراکز پزشکی دانشگاهی بزرگی محدود شده. می‌خواستم نظرتون رو بدونم راجع.
That's a keen observation. I think the development of this technology has been hard, and it's very expensive. I think right now the ability to do it requires a lot of financial support and a lot of expertise. And you can think of it like PET, but in the 1970s. You needed a cyclotron to do PET. And I think the reason why everyone can do PET now is because you don't actually need a cyclotron to make FDG. You can buy FDG. You don't need this both a nuclear reactor and a physicist who can hit this target and all of these different things to be able to just conduct traditional PET studies, right? You just need a pharmacy technician to make a bottle, make a syringe, and they can inject people all day long with FDG and buy a PET scanner from Siemens or GE or whoever. There's like a million of these people making clinical PET scanners, and so but if you look at PET in the 1970s, there weren't even that many places. And so I think we are very early in the game in this field. But the benefit is that because we're dealing with something non-radioactive, because it's attached to a detector which already exists, meaning MRI. So at the time for PET, they didn't have, you need a nuclear reactor and they didn't have the PET scanner. So it took a very long time for them to develop what was needed, even though we knew that it was a good idea. And so I think here we have a leg up. The startup world, as well as the big companies, have recognized that they need to make this cheaper, faster. And I think a lot of the exciting developments are going to come in that space. Is that if we can make these tools sit on a desktop and, adjacent to every MRI machine, then you will see a massive increase in how much is used. I would expect much more than PET, because there are many more MRI scanners. The onus on the person is much lower. You don't need to do radioactivity. So I think in the clinical workflow, it will also become more useful. So I think for the clinician, if such a technique worked and it was scalable, it would be a routinely used thing. The same way that people use fluoroscopy. They're not even radiologists. Or you do EKG. I mean, I think there's just tons of these applications where this could also play a role. So it's just early in the development, but we're actually very close to becoming exponential, I would say.
خیلی ممنون. من مجددا به آدینس عزیز عرض میکنم اگر که دوستان این سوال بپرسین، هم میتونین در واقع در اسلایدو وارد کنید یا به استیج بیاین یا اینکه بک چنل به دوستان بفرستین. سوالی که داشتم از شما در مورد در واقع مطالعه همین روش برای سرطان‌های مختلف هستش. شما روی پراستیت کار کردید، روی در واقع برین کار کردید. اینکه چه تفاوتی دارن و اینکه آیا اینجوری هست که یکی از اینا بیشتر چالش برانگیز باشه بررسی و تحقیق در موردشون؟ اگه در مورد این انواع مختلف و اینکه کاربرد HPMRI تو هر کدوم توضیح بدین و چالش‌هایی که دارن ممنون میشه.
Sure. Generally, you could think of this approach for all cancers being that cancers generally upregulate metabolism, glycolytic metabolism, to drive growth. I mean, cancers are one of the kind of major hallmarks of cancer is the kind of unlocked proliferation of a cell. I mean, it's like the definition of cancer. To facilitate that, you need to upregulate your metabolism. And the kind of major pathway that does that is glycolysis. And this molecule that we've translated, pyruvate, reads out the last step in glycolysis. So in the setting of prostate cancer, it appears to be very useful in grading prostate cancer. So in the setting of prostate cancer, that is a big question. If many of you, if you're older, let's say over 50, or your father or grandfather gets to that age where they, start doing routine prostate examinations and PSA tests and whatever, you find that the major question that gets asked before surgery is, does this person need surgery? And it's amazing that question is still hard to answer. We do the traditional test that's done is they take a endorectal probe, so a probe that goes, in your rectum, right to the prostate, and takes 12 biopsy pores from this very small gland. So prostate gland is about 4 centimeters in diameter, and they take 12 biopsies from it, needle aspirates, from what's called a sextant, 6 regions, and 2 from each region, and try to diagnose whether or not you have prostate cancer from that, this sampling. And you can imagine immediately, if you're a scientist, this is always going to miss. It will always miss. There's always tons of the gland that you won't see. And additionally, depending on how you sample, will give you a skewed perspective on what's there. So non-invasive techniques should be able to address this. But even then, the question is this aggressive enough that this person should get surgery? Or should we not do anything? What's called watchful waiting or active surveillance, or just kind of go back and look every day. And, this question, which hopefully we can address with this technology, could change how we deal with prostate cancer. More people will probably not have surgery. Or maybe the people that do have surgery, we can catch them earlier, because the kind of side issues associated with surgery are actually pretty profound. I mean, there are incontinence and lack of sexual drive for some 5% of people, and other issues. And so you can imagine having surgery is not something you want to do unless you absolutely have to. In the setting of prostate cancer, at least primary prostate cancer, that is the question: how aggressive is it? And we think that metabolism may be a way of doing kind of addressing that and trying to characterize it non-invasively on the whole gland scale. So in terms of 3D imaging of the entire gland. In the setting of brain tumors, the question is a little bit different. We know you have a brain tumor. We don't need this technology to find the brain tumor. I think there's a common misconception of cancer imaging in general, which is we actually do a pretty good job of finding tumors. Our problem is we don't understand them. Like, we see there's something there. Maybe we don't know it's a cancer, but we think it's a cancer. But we don't know what kind of cancer it is. We don't know if it's going to kill you. We don't know how fast it's growing. We don't know if it's responding to therapy. So like in the setting of brain tumors, we give therapies. We have a very short time window. And our question is, did we do enough to kill this tumor? Do we do surgery, radiation, chemo afterwards. Is this tumor responding, or is it actually still growing? And because we don't have time to wait for the tumor size to change, which takes a long time, so by the time the tumor is getting bigger, the show is over. Like you can't do anything about it for these patients. And so our hope is in the setting of imaging patients that have, for example, resected disease, meaning like you did surgery and took the tumor out, and there's some maybe tissue left that maybe regrows a tumor. Can we image that early? Can we take that person back to surgery? Can we do something else for that person? Because the quality of life aspects of these therapies is very bad, especially in the setting of kids. Like where for a lot of these tumors, they live maybe 9 months post-diagnosis. Once they find there's a tumor, between every therapy that we have, they still pass away. And so I think it's in trying to understand therapy response in brain tumors. So it's a different. I think in every one of these different applications, there is a role for this kind of metabolic imaging to tell you something different that we don't fully understand, and that there's just a lot we don't know.
خیلی متشکرم کیوان عزیز. من یه سوال، دو تا سوال داشتم، یه سوال متفاوت و یه فالوآپ از صحبتی که همین الان کردید. سوال اصلیم این بود که یه کار اخیری انجام دادید توی کارهای خودتون به اسم Hyperpolarized NMR پلتفرم و همچنین فانکشنالیتی اون. میخواستم در مورد اون یکم توضیح بدید. و سوال فالوآپ هم که از این موضوع اخیری که گفتین اینه که ایموشنالی چجوری میتونید تو اون مرکز کار کنید وقتی که با افرادی سر کار دارید که بسیار توی لول حساسی از زندگی هستن و به ویژه کودکانی که الان مثال زدید.
Sure. So the hyperpolarized micro NMR is easier. So that I think is a very simple extension of an idea that isn't ours, which is like I said, NMR is very insensitive. Hyperpolarized NMR overcomes some of that insensitivity, but another property of NMR or magnetic resonance in general is the way we detect it. And we're very interested in other ways of detecting magnetic resonance. And we have other projects in the lab studying things like optically detected magnetic resonance and other things. But traditional electrical sensing of NMR is kind of like a transformer. You have a coil of wire tuned to a frequency. That frequency is matched to the frequency of your processing nuclear spins. And so, the most simple form of an NMR detector is a solenoid coil wrapped around a tube. And this is like the kind of original idea for an NMR detector. And so we know that as the coil gets smaller, you have this property called the filling factor. And so your number of spins relative to the volume goes up, and so your sensitivity goes up. And there's a limit to this, but the idea is that as you make the detector smaller, you increase your sensitivity for the same number of spins. And our idea was, well, we would like to approach single-cell NMR, non-invasive detection of single-cell metabolic flux, which is something challenging to do, basically to image the conversion of a substrate to products, but in a single cell. So we published a paper in Science Advances a number of years ago demonstrating this kind of first micro NMR platform with hyperpolarization. So we could take advantage of some of the microfluidics people have developed, in chemie for trying to introduce, substrates to a reactor. And we combine that with a microfabricated solenoid type of coil inside of a permanent magnet. And so the idea there is that we can visualize metabolism in small numbers of cells. So in that platform, we got down to in the thousands of cells, which still is about, I think the competing technology for measuring flux, aside from optical methods, because in optical methods, you need a dye. You're not really measuring the substrate and the products. You're measuring surrogates. But if you were to compare it to something where you actually measure the analyte, it would be mass spec, probably. And there are studies of people doing single cell mass spec, but every time you do a measurement, you destroy the sample. So you cannot measure a flux in the same sample because you only take one time point. So you have to take many, many samples. But, typically those isotope tracing studies for, mass spec are done with, in a normal lab are done with, millions of cells. And that is what it would take to measure a rate. We can measure it in that platform. We would measure it in about 10,000 cells, non-invasively, and then you can take those cells and you can do other things with them, RNA-seq, whatever. But more recently, we've been working on a new platform in collaboration with some colleagues in Germany, which we're hoping to see if we can get to work, which is a nanoscale coil. And in that setting, we do think we have the potential of reaching, kind of a nanoliter volume. And in a nanoliter, we're really close to, single cell resolution. So I think the next version of that system may actually approach kind of our ultimate goal, which is this kind of high throughput, single or few cell kind of situation. But that's the reason for developing that platform and kind of why. And so once we have such a thing, we can start to ask questions about flux in small numbers of cells, like stem cells or circulating tumor cells, or, in the application T cells. We're very interested in immune cells. And so I think there's, I have a number of students in the lab that are working on kind of different aspects of this, both like the engineering and kind of physics side, as well as like developing probes. So it's an area that we're very interested in kind of pursuing more. On your second question, it is very hard, personally, to work in a place where people are, going through cancer treatment. But I would say that what's actually harder is meeting people, family members, friends, who have someone who either like they have cancer or their child has cancer, and you go through that process with them. You bring them to Sloan. That's when you really experience it. It becomes more. It's easier when it's somebody else that you don't know. You can be objective. We can have patients come in and image them, and it's okay because you're not 100% involved. But I've experienced people in my family passing away from cancer recently, as well as close friends whose child passed away from a brain tumor. It's different when it's your family. It's different when you try to bring them into your world and you see it, and it's very hard. It's very hard to be objective, but you try to balance that with, I could be working at a traditional university, like I could go back to Berkeley or Stanford or something. But if you're in the engineering school or the biochemistry department or whatever, you are separated from that application. You do kind of very basic or maybe even irrelevant things that 100 years from now no one will say is like useful. In this setting, maybe you do something that dramatically has an effect instantly. You image a person, inject something in a person, maybe it helps them. So you have that closeness to some utility, and so you can see that, and I think that helps overcome the emotional, sad, challenging part of seeing people going through probably the hardest point of their lives. I mean, this is probably the hardest thing that any of these people that come to Sloan Kettering are dealing with, that will ever deal with. And when they get better, they leave. You don't see them anymore. So I think that aspect makes it challenging. I will say on the kids thing, which I think is amazing, one of the most inspiring things I've seen at Sloan Kettering, we have a very large pediatric oncology program. I think it's been named MSK Kids, and one of the kind of glimmering light best things that have happened is this dramatic attention and kind of curative direction for a small kind of brain tumor, neuroblastoma, where an antibody was developed at Sloan Kettering about 30 years ago called AH9, which eventually was labeled with a radioactive substrate and has become a company which has done very well financially, called YMABS, as they labeled the yttrium. So this company, well, with collaboration with a company, but mostly through Sloan Kettering, started to inject people with this antibody and cure these kids. And so, every year they have a kind of a reunion for these kids that they treated, who were like, let's say 4 or 5 years old. And they come back and they've graduated from high school and they go on to college, and you see these people get married, and it's like they're living normal lives. And so there are these success stories from people that would have died, kids that never would have gone to elementary school, that you see walking around. So I think those are the things that keep everyone positive, but it is a very challenging thing to do.
خیلی از پاسخ خوبتون ممنونم در هر دو بخش. بر من خیلی آی اوپنینگ بود، خیلی استفاده کردم. متشکرم.
خیلی هم عالی. واقعا خیلی من دارم یاد میگیرم و خیلی لذت میبرم از بحثایی که تا این لحظه شد. حالا خب من رشتم بیزینس هست و کلا یه خورده دور هستم از این تاپیکی که شما دارین راجعش صحبت میکنین. منتها برای من خیلی سوال بود راجع به این ایمیجینگ متد که بدونم که این رو شما فقط میتونید در مورد سرطان استفاده کنید یا اینکه نه مثلا این اسپیلوور هست از یک فعالیت دیگه، یه ریسرچ دیگه که الان توی سرطان هم یعنی توی دتکشن سرطان هم کاربرد داره. خوشحال میشم بدونم اپلیکیشن دیگه که این ایمیجینگ متد داره چجوری هستش و اینکه چقدر تیلور شده فقط برای سرطان.
So it's I would say it's very kind of general. You could imagine approaching it like all of nuclear medicine, in that we can infuse substrates for any metabolic reaction, presumably. I mean, this requires development, but many metabolic reactions, and just see what they get converted to, how fast and where. So it has applications outside of cancer. Many, actually, we have studied many on diabetes, fatty liver disease, normal aging, neurodegenerative diseases, all these different things in different parts of your body. In terms of like it being tailored for cancer, it's more in the regime of we designed molecules, or we are not even designing, let's say tracing molecules that cancer uses more. And in that way it is tailored for cancer, maybe. But it's not, we're not changing the system. We're just asking what it's doing. And we understand how cancer in some respects requires certain things. We're learning more about this every day. You could imagine, metabolite A is used by cancers to do, this biochemistry that is important. And so people develop drugs for that biochemistry. And also we want to understand if that drug stops that molecule from getting metabolized in the cancer cell. And so you could think of this imaging as, well, we image that molecule and where it goes, and does it turn into the product? And that gives you a readout of that biochemistry, which is important to cancer. But it also could be important to diabetes. I mean, we're very interested in trying to study the pathology of diabetes. And, Sloan Kettering is kind of a funny place because it's actually not Memorial Sloan Kettering or Memorial Hospital for cancer. It's actually Memorial Hospital for cancer and other allied diseases. And the reason is because many people with cancer have many other problems. They tend to have other comorbidities. And also, there's a kind of interesting side kind of curiosity, which is that many people who get cancer don't typically develop neurodegenerative diseases. And many people that develop neurodegenerative diseases or have a familial history for that, so say Parkinson's, Alzheimer's, whatever, tend to not get cancer. It's kind of a weird curiosity. And so actually many people at Sloan Kettering study development and study those diseases, Alzheimer's, Parkinson's, to try to understand also what is different about them relative to cancer. And so we are, in a lot of ways, the institution is very general, but also a lot of the things that we develop are not necessarily just cancer specific. They have many applications outside of that. And I think outside of Sloan Kettering, a lot of people use these tools for other things as well, that are not cancer specific.
مرسی، خیلی ممنون کیوان عزیز از توضیحاتتون. ما یه سوال از اسلایدو هم داریم، اما قبل از اون افت جان به شما خوش آمد میگیم و ممنون که تا این لحظه منتظر بودی. خوشحال میشیم که سوال شما رو بشنویم. افت جان هستید؟
سلام علیکم.
سلام.
خیلی ممنون از زحمتی که کشیدین برای این اتاق خوب و آقای دکتر کیوان کشاری خیلی ممنون از صحبتاتون، من خیلی لذت بردم. فقط واقعیتش اینه که چون به اصطلاح بیسیک ساینس این کار رو نمیدونم، من بیشتر مولکولار بایولوژیستم و کارام توی دراگ دیولوپمنته، در حالی که بخش بایوکمیستری شو تا یه حدودی میفهمم، ولی خب بخشای انجینیرینگش نه متاسفانه. من سوالم دو تا هست. یکی این که آیا خود دارو مثلا فرض کنید ما الان داریم یه پپتاید رو دیولوپ میکنیم برای تریتمنت چند نوع کنسر خاص، اختصاصا تمرکزمون روی به اصطلاح لوکال کنسر تراپی هست برای این کنسرهای خاص که یکیشون هم همون جی بی ام هست، تومور مغز، که خب الان خیلی روش داره کار میشه. میخوام ببینم که آیا این متد کاربردی میتونه داشته باشه برای در واقع تریس کردن خود به اصطلاح API، خود اکتیو مولکول توی ساید آف د تومور. یعنی اگه که ما بخوایم داخل تومور به اصطلاح بایو دیستریبیشن و متابولایت فورمیشن یه دارویی که پروتئین بیسد هست، مثلا یه پپتاید بیسد دراگ یا همونطور انتیبادی رو مثال زدین، میخوام ببینم که با چجوری میشه استفاده کرد، اصلا کاربردی داره و اگه کاربردی داره اون بیسیک مکانیزمش چیه؟ آیا فقط لیبل شدن خود تراپیوتیکه یا اینکه کارهای دیگه شما انجام میدین برای به اصطلاح ویژوالیزیشن؟ سوال دوم هست اینه که آیا اصلا کاربردی میتونه توی خود دلیوری اینتراتومورالی داشته باشه؟ مثلا روش های دیگه‌ای که الان هست برای لوکال کانسر تراپی، حالا من نمیخوام اسم ببرم ولی میخوام برم آیا این روش اختصاصا میتونه کاربردی داشته باشه به خصوص برای داروهایی که کندید خیلی خوبی هست، مثل داروی خودمون کندید خیلی خوبیه برای لوکال کانسر تراپی، به خاطر اینکه یه سل پنتریتینگ پپتایده، خودش وارد سلول میشه. اگه لطف کنین جواب بدین خیلی ممنون میشم. متشکرم.
Sure. So, your first question, if you want to, so okay, peptides and proteins tend to be larger than the molecules that we inject. However, if you are interested in what these peptides being accumulated in a tumor, the best strategy is actually to radio label them with a nuclear agent. So when we do some of that in the lab, so where you don't use hyperpolarization, you actually would use PET. You can put gallium-68 or one of these radio tracers on it, and or copper-64. There are like many different radio metals that you can use, and you would image it with PET. That would be the easiest to see accumulation of a peptide. Would be much more sensitive. In terms of direct injection to a tumor, which I think is what you mean, there's a lot of ways to visualize that peptide being there. Again, you can do it radioactive. There are gadolinium-based probes for MRI. You can put on the molecule, you can label it. It just depends on what you want to do your imaging with. For what we do, we tend to not hyperpolarize the drug because you inject very low concentrations of the drug usually. What we tend to image is the product of the reaction, meaning so say your peptide turns something off, some biochemistry off. We try to image that biochemistry, whatever that reaction is, that other thing, or what the end product is. And that tends to be more important than just do you make your active metabolite, because you could make the active metabolite and the cell doesn't care. That's I think the most kind of, but the biggest problem in that aspect of drug this kind of action in vivo. In terms of delivery, I would say that actually nuclear methods would probably be better for you. And those are very easy. That chemistry is easy. Most of it is, some chelator is put on your peptide. They're like standard. You can just buy it. And then, have it labeled by somebody who does PET. It's not hard to do. In terms of, but drug action. So I think for PET people, it's very hard to image drug action. The drug actually does something. And for us, that is kind of the place where I think this technology would be useful. So your peptide stops glycolysis from happening. We image the downstream product of that specific enzymatic reaction. So an example of that for, just recently we've done. In cancer, people are interested, for example, in gliomas or in leukemia. They're interested in this enzyme called IDH, which is a TCA cycle enzyme that converts interconverts isocitrate and alpha-ketoglutarate. So it generates alpha-ketoglutarate in the TCA cycle, if you remember that from kind of biochemistry 1. But when it's mutated, it creates a oncometabolite called 2-hydroxyglutarate. This is kind of a dead-end product. And people have developed drugs, peptides, a lot of small molecules to target that reaction, to stop it from happening. And so we don't try to follow the drug. We try to ask, does the cell stop making 2-HG? And so we make substrates that go into that reaction, and then you can image the results. We can image the formation of 2-HG. And if that goes away, then that means your drug works. It got to the target, and at the target, it was effective. So I think that's how you would probably use this technology in that space. Another is just generally, if you image a process which is related to cell death. So if the cell dies, then this reaction happens. Then you can look at just general effectiveness. And there are molecules for that as well. So I think that's the way that you would approach that. In terms of like local delivery, again, I think the easiest, and the example I gave before of this direction called that YMABs took, where they, with an antibody they wanted to image, they actually, the original version of this, they still do this, is they take a very, very long catheter, like an extremely long, thin catheter, and the neurosurgeon directly places it in the child's brain tumor. So it's like right in the middle of the child's brain. And they directly inject using, just like what's called convection injection. They directly inject into the tumor radioactive, it's like iodinated antibody, actually, the original version, directly into the patient's brain. And they image it also, SPECT. So they do SPECT imaging for delivery and also radioactive therapy treatment at the same time. And it's extremely successful. So, I mean, I think radioactive tracing would be a better solution for your drug delivery problem. But drug action would maybe be more of a hyperpolarized problem.
خیلی ممنون، متشکرم. به خصوص اون CED رو در واقع ما خودمون داریم روش کار میکنیم و در مورد نوروبلاستوما ظاهرا شما این کار یا اون کامپانی این کارو کرده. ما در مورد گیلیوبلاستوما داریم این کارو میکنیم. البته همکارا توی UCSF هستن، ولی من اصلا نمیدونستم که توی اسلون هم این کار میشه. من حال اگه مشکلی نیست من با ایمیل باهاتون فالوآپ میکنم که وقت این گروه بیشتر نگیره. خیلی ممنون، متشکرم.
بسیار عالی، مرسی از شما. یکی از سوالهای اسلایدر رو میپرسیم. دوستان آدینس هم اگر دوست داشتن سوالشون رو از خود کیوان عزیز بپرسن، میتونید تشریف بیارید به استیج، یا اینکه سوالتون رو در اسلایدو، همین لینکی که رو اکانت اینفو هست، در واقع وارد کنید. حدود 1 ساعت هست که کیوان عزیز مهمان کلاب ایرانیان هاروارد هستن. حالا برای کسایی که تازه وارد اتاق شدن، ایشون عضو هیئت علمی در مرکز سرطان مموریال اسلون کترینگ هستن و در ارتباط با متابولیسم سرطان، تشخیص و درمان با استفاده از روش‌های غیر تهاجمی و تغییرات متابولیک کار میکنن و روش تصویربرداری هم که استفاده میکنن Hyperpolarized MRI هستش. یه سوالی که تو اسلایدو پرسیدن رو دوست داشتم مطرح کنم اینجا، که پرسیدن که آیا میتونید از در واقع اون سیرکولیتینگ تومور سل ها، در واقع سلول هایی که از اون تومورها جدا میشن و در بدن در جریان هستن، بیایید از این در واقع روش هایپرپولاریزیشن استفاده کنید تا اطلاعاتی، دانش بیشتری در مورد کنسر متاستاز به دست بیارید. اگر این سوالم پاسخ بدید من.
Sure. It's a great question. We would absolutely like to do that. Circulating tumor cells have kind of two general benefits for us. One is just for cancer detection. I think optical methods are good for telling you certain properties of cells, but I think metabolism, if we can non-invasively monitor it in single cells, provides another angle to look for cancer, sort for cancer stem cells. So the big problem with circulating cells is that many of the cells in your circulation are not cancer. So filtering out the ones that could be cancer and then trying to analyze those is challenging. So one, we would like to be able to analyze small numbers of cells and ask which ones are cancer and then what are the properties of those. And I think that is one of the directions of trying to develop a more sensitive nano platform in combination with hyperpolarized probes to do that. So right now we don't have the sensitivity to see single cells directly with our methods. I mean, you can with other methods, but we don't have the ability to do it with ours yet. But it is something we're trying to develop, both to see if we can visualize cancer circulating tumor cells and also to better understand them. So once you do visualize them, what can we say about their metabolism that's different? And then if we were to treat them specifically, how would they behave? Because many of the therapies we develop are against a primary lesion or a metastatic lesion, but in reality, metastases come from these circulating tumor cells. They must have some properties that allow them to survive transport from the primary lesion to make metastatic sites. And so being able to understand circulating tumor cells better would tell us a lot more about the generation of metastases and how to address them. So there's a lot of reasons for wanting to do that, and that is exactly the reason why we're trying to develop more sensitive ways of looking at single cells.
مرسی، خیلی ممنون. حالا من یه سوال فالوآپی اینجا داشتم و این بود که آیا این تشخیص‌های جدید یا درمان‌های جدید دیگه‌ای هم وجود داره که در واقع در چند سال آینده بر اساس همین اکتشافات متابولیسم سرطان که امروز اتفاق میفته ما بتونیم ببینیم؟ چون من نمیدونم که حالا مثلا با این اکتشافات اخیر در مورد همین بحث متابولیسم سرطان که خیلی تعداد زیادی هم وجود داره، بحث یعنی مطالعه روش میشه، هنوز روش تشخیص و درمان بیماران مبتلا به سرطان رو تغییر داده یا نه؟ مرسی.
Sure. I would say we have made progress. Targeting cancer metabolism has started to become a direction that many people are pursuing. I think visualizing metabolism allows us a way of seeing if that works or if it matters. But more recently, there have been a lot more metabolic inhibitors that have been translated for treating different cancers in combination with things like immunotherapy. We think one of the reasons why immunotherapy doesn't work is because of cancer and immune cell metabolism. So I think maybe in the next 5, maybe more like 10 years, there will be some major advances in this space. We will learn a lot more about cancer metabolism, and maybe that will be something that will help us better address this disease. So I would say there are a lot of things that will happen in the future and things that we are. It's interesting about, I would say when as a society, we were a scientific kind of society, we were interested in metabolism. It was, kind of the early 1900s to the 1950s. And then we stopped, there was about 13 Nobel Prizes that were given out for what's called intermediary metabolism. Basically, all of the metabolic reactions that you study in, biochemistry, biology textbooks, those were discovered in like the first 50 years of the 20th century. But then we stopped caring about it for about 50 years. We were interested in genetics, then proteomics, and actually until this last, I would say 15 to 20 years, there's been a resurgence in being interested in cancer metabolism. And I think that has spurred an interest in thinking about better drugs to specifically target these metabolic processes that we know cancer cells. So we're learning about new cancer metabolism every day, and we're using that to develop new diagnostic tools and new drugs every day. So it will just get better. So hopefully we'll make a dent in some of these diseases as a result of some of these metabolic directions.
بله مرسی. من هم امیدوارم که این اتفاق بیفته و ممنون از توضیحات خوبتون و دیدگاهتون راجع به در واقع پیش بینی اینکه سرطان ممکنه که با این روش‌های که شما کار میکنید این در واقع با ترکیب شدن با روش‌های دیگه بتونه یک قدم خیلی مفیدی رو تو این زمینه داشته باشه. یه سوالی که توی اسلایدو از شما پرسیدن اینه که میگن که چقدر همکاری‌های بین‌المللی در واقع اینترنشنال کلابوریشن توی تحقیقات سرطان موثر بوده و چه همکاری‌های متمایز و خاصی توی این زمینه وجود داره؟
We collaborate extensively, I would say, nationally and internationally, all over. We're happy to work with anybody who has an interesting question that we can do something about. Internally, there are just so many questions. I think, it's not hard to find a good cancer question. In terms of, outside this, I mean, the field is huge. So I think if you use kind of the National Institutes of Health as like an argument, the NCI is like by far one of the biggest funders of research. And, that's really kind of, I think, gauges the interest in cancer generally. We don't have a problem with, I think, finding collaborators. I think there is generally like a big interest in this kind of research. And so if you are interested in cancer, or you're interested in biochemistry, or you're interested in engineering, I mean, I think there's a home for you to think about these things, kind of in this space as well. So it's actually quite large. We kind of tend to collaborate with people who are not like us. And I think that is both challenging and, motivating. It's challenging because sometimes it's hard to speak the other person's language or understand what they're interested in. Maybe you're interested in slightly different things, but it's motivating because you can try to address problems in other fields. And so we tend to collaborate a lot. And we're, nowadays with Zoom and, and WebEx and whatever, you don't just have to be local. So it's easy to collaborate outside of just, what's happening here.
خیلی ممنون، مرسی از پاسختون. یه سوالی که تو اسلایدو پرسیدن اینه که فکرم دو تا سوال مشابه هم هست. آیا از این روش در واقع همون MRI برای تارگت قرار دادن متابولیسم کانسر استم سل ها هم استفاده کرد؟
So it's yes on some level. I think it depends on the metabolic process and cancer stem cells in general. So I think if we can identify a process which is specific to cancer stem cells, and there are some, or that is important to cancer stem cells, then following the metabolism of those is possible because you have that level of specificity. I think additionally, if we can sort stem cells, and this is something that we did in a previous paper, actually in that first micro NMR paper, we actually took some mouse models of leukemia and then sorted, stem cell populations and then asked, could we analyze the metabolism of those cells? That's kind of one of the reasons why we built that platform, is to increase our sensitivity, because there aren't that many cancer stem cells or stem-like populations in a given tumor. So we could do those experiments, and we can learn a lot about. So, like for example, just like anecdotal, simple thing from that paper is we learned that leukemia stem cells are more glycolytic than their progenitor cancer cells, or let's say progenitor stem cells. And so that tells us something about the reliance of cancer stem cells on glycolytic metabolism versus, how much we appreciate of their, dedifferentiated or differentiated cancer progeny. So I think you can learn a lot about cancer stem cells. Now, whether or not we can visualize them in vivo becomes a function of what their metabolism is and how much they do it. That kind of dictates our sensitivity. But it is something we are really interested in because plays a major role in the biology of the tumor. So that's the way that I would approach it. And that's the way we've thought about it in the lab.
بسیار عالی. حالا تو این پروسه‌ای که شما حالا میایید از این تصویر برداری استفاده میکنید، آیا تا به حال از روش‌های مثل هوش مصنوعی استفاده کردید، از توانایی‌هاشون که بیاد یاد بگیره از این در واقع مالتی مودال دیتا ها و کمک کنه به اون بحث سنسیتیویتی روشتون و حالا اینکه اطلاعات بهتری رو بتونید به دست بیارید از اون واکنش‌هایی که داخل اون سلول‌های سرطان اتفاق میفته؟ آیا خودتون انجام دادید یا میدونید که ریسرچ در این زمینه انجام میشه؟
We are kind of just scratching the surface of that. I think that once we generate larger data sets of images of people using this method, it lends itself to all of those high dimensional data kind of directions, machine learning, deep learning. I think in the clinical space, probably classified all as like radiomics, but this idea of adding this data set in we believe would dramatically increase the ability of these machine learning algorithms to be more successful. I think we just need to generate more data, and the data needs to be robust. I think the data generated different places needs to be standardized, and kind of all the same problems that are for every other imaging approach. So اگه میتونیم problem رو address کنیم, we will have like a way of incorporating this data into those and hopefully making those algorithms work better. But yeah, it would easily lend itself to that. But we haven't done it yet. I mean, we just don't. I think we don't have enough data in terms of data set sizes, and we're in the process of trying to enhance our resolution. If we can get better spatial resolution, match closer to structural imaging. So, for example, what is normally used for this kind of data is CT or MRI, with kind of higher traditional MRI with higher spatial resolution. I think if we can get to higher spatial resolution, we will be even better at being incorporated into these. Similar to what's done with functional MRI or diffusion MRI or contrast enhanced CT, like the way that those are incorporated as well into these machine learning algorithms. I think we would also be part of that process.
خیلی ممنون. حالا میدونم که منابع یکی از سالهایی که پرسن در مورد همین منابع هست که میتونن بیشتر در مورد سرطان ها یا دوام سرطان یاد بگیرن. مطمئنا منابع که خیلی زیاد هست، چه در یوتیوب چه در اینترنت. اما حالا سوالی که پرسیدم برای کسی که در واقع دوست داره که در مورد سرطان برای علاقه شخصی یاد بگیره، آیا شما کتابی یا پادکستی رو معرفی میکنید؟ مرسی.
I'm not really a podcast person, but I feel like a lot of if you're trying to learn from textbooks these days, I feel like you're maybe behind. It's just the fields are moving so fast that the only way I see to kind of catch these fields, to be in them, to understand them, is to read reviews. And a lot of people write these, kind of summary reviews of state of fields. For example, in the setting of GBM treatment or research, for example, in Nature reviews neuroscience, or Nature Cancer puts out a lot of these. I think actually those tend to be much more informative. They're shorter, and also the bar may be hard to reach if you want to get a general understanding at the beginning, but once you overcome that, it tends to be much more useful than trying to look for these kind of general audience textbooks. So you would, or even just books, like you can read these books, but I don't think they'll be as effective if you're really interested in trying to understand what's going on. These reviews tend to be more so. I would follow, and I do actually, we use Slack in my group a lot, and we've set it up so that these RSS feeds kick to different channels in our Slack. And I think that helps the students learn very rapidly, kind of, you just get forced with tons and tons of knowledge constantly, just like kind of in your face all the time, and some of it is like a fire hose. what they say, drinking from a fire hose. You won't get a lot of the water, but some of it will go in. And I think, over time you will learn more about it. So my suggestion would be that, in terms of wanting to learn more about these kind of general reviews.
بسیار عالی، مرسی کیوان عزیز. یه سوالی در مورد مرکزتون داشتم و حالا هزینه های تحقیقاتی که انجام میشه در مقایسه با دانشگاه های دیگه چه تفاوتی داره؟ در واقع چقدر از اون گرانت هایی که شما میگیرید برای ریسرچ، پولی که برای ریسرچ استفاده میکنید از گرانت ها میاد، از جاهای مثل NIH، و چقدرش از دونیشن هستش، و اینکه آیا تفاوتی هم با دانشگاهی دیگه داره یا نه، مثل تمام جاها کلاسیک هستش و بیشتر بعد از طریق گرانت شما فاند.
So in some ways we're very similar. So my experience has been with many kind of public universities and private places, and so I would say in terms of my group, we are we get support from federal grants, so mostly federal grants. So it's the NIH, or I don't currently, but we have in the past DOD, DOE, other federal kind of funding agencies. Not as much NSF, because the NSF doesn't like to fund us. They like to fund kind of even more basic science. I mean, we get them every once in a while, but we're like mostly NIH in terms of federal grants. And then a large proportion of foundation grants. There's quite a few of these foundations that fund cancer research and tend to fund us. And then, another percentage of philanthropy. I mean, we have Sloan Kettering does a very good job raising money, raising awareness. So we have a percentage, I think, of probably the funding of my lab over the years has been through internal funding from philanthropy, but so I think we have maybe a little bit more of the philanthropy and foundations than maybe say you would have like a Harvard or Penn, but I mean it's just there's still a lot of focus on grants, I think, to fund the research in the lab. We have the benefit of being a private and fairly well-funded institution, so we have very strong core facilities. And that helps us quite a lot. I mean, it's funded by our cancer center grant, and we're not a big place, so it's easier for, I think, them to balance the budget. I think if we were a bigger place with more things going on, it would be we would be open to a lot of things changing and a lot of demand. So in some ways, a lot of people are thinking in the same space. So when we buy equipment, everyone's kind of on board because they're all generally in the same space. They're not competing with, say, what the neurologists are very interested in today, or the cardiologists, or the solid state physicists are interested in, because we don't have all these other departments. Everyone's kind of generally focused in the same space, so they have a similar interest in a lot of the same things. So that kind of unionization, or kind of uniting of things, is helpful. So it allows us to be smaller and nimble, and also more financially kind of solid. I think it's easier in some ways. We get a lot of internal support to run the lab, so I think that makes life a little bit easier. But there is this is the same. You want to make associate. The requirements for making associate are the same. They are at MIT or at Penn or whatever, or to make full professor. And I think it's not that different. It's similar, I would say.
خیلی ممنون، مرسی از توضیحاتتون دکتر کیوان عزیز. یه سوالی که از من توی بک چنل پرسیدن اینه که یک روز معمولی آقای دکتر چطور میگذره؟ اگه تمایل داشتید میتونید از اون دیلی روتین روزانه‌تون برای یک روز معمولی بگید که چطور میگذره.
So the funny question, I think, if you ask my wife, I'm sure she has a different opinion of what my day is like. But it's a lot of meetings, more meetings than I want to have. always. But I try to meet with everyone in my lab at one-on-one at least once every 2 weeks. That's kind of a minimum thing. So especially when I'm not traveling, I try to like hold to that schedule, because not because I don't think that they're doing something, or that I think the issue is more that sometimes you need help and you don't know it. And I always felt like you could have a conversation for 20 minutes, and if it's like, okay, everything is great and I figured this out, I don't understand this, can we talk about this, and whatever, it's fine. But sometimes you don't realize that you're kind of going down the rabbit hole in a bad place, and you're going to be stuck there for 6 months, and maybe I can help you not go to that hole. So I think there is a kind of a need for that. So I have a tendency of meeting with people often. I hate writing grants. I dislike kind of being in too many meetings. I get very tired reviewing papers. So in the middle of all of these things, I have a tendency of just kind of walking around in the lab, which I'm sure drives people in my lab insane, because they will be doing something and then I will randomly show up and waste a bunch of their time talking to them about nonsense. But I like going in the lab. It makes me feel like I'm still doing things. It's on some level I live vicariously through their science. Like I don't get to do anything useful. So whenever I get a chance, especially if I'm in town, I tend to try to go in the lab and do things. But in a given day, I probably do, like, especially now with Zoom, which is horrible. I hate Zoom. I'm probably like 70% in meetings. I don't have to teach that much, so I'm not really teaching right now. But mostly kind of meetings, high level meetings, and meetings with people in my lab, and then, a lot of writing. And I drink a lot of coffee, maybe too much coffee. That's my other problem.
بسیار عالی، مرسی از شما. جالب بود برامون. یه سوالی که داشتم در مورد اون رگولیشن هایی که هستش در مورد تحقیقات، حالا اسپسیفیکلی در مورد کنسر تو آمریکا، فرض کنید اون محدودیت هایی که حالا FDA داره، مقرراتی که FDA نیاز داره که اون ریسرچرها فالو کنن، ها چه فاندینگ ایجنسی، و تفاوتش با کشورهای دیگه، و اینکه چقدر این رگولیشن ها میتونه تاثیر بذاره رو پیس در واقع تحقیقات شما در مورد کنسر. ممنون میشم که اگر کامنتی دارید.
So I think, unlike other diseases, which are kind of long term, maybe not as kind of rapid onset, the bar becomes a little bit lower for cancer therapeutics, I would say. Meaning that if you have something, a drug, which is effective in model systems and has some low level toxicity, you can get into a phase 0, phase 1 clinical trial pretty rapidly, because I think the FDA and the kind of a lot of companies are interested to finance these, because any pushing of the needle becomes very impactful. Unlike, I think it's much more challenging in the setting of, if you were to develop a new drug for diabetes or something, or heart disease, it's, the safety levels and the testing of those things needs to be much higher, because you're treating people who are otherwise healthy. I think in the setting of cancer, this person, especially for some of these brain tumors, pancreatic cancer, their 5-year survival is 18 months, maybe less. So we have a very big need for developing those therapies. So I think in the US, there is a general push always for trying to translate new and exciting, potentially impactful strategies. I mean, you could think of it as similar to COVID. Like some of these therapies that we're doing for COVID got into people extremely fast. I mean, think about how fast you got this mRNA vaccine. I'm sure many of you have been vaccinated. I hope all of you. But, from inception to injection into like millions and millions of people was super fast. But that's because we had a dramatic need. People were dying. And I think if you have early onset disease where you can make the argument that this is a population that needs it, the FDA is not completely standing in your way. Outside the US, I don't have a lot of experience in Asia, virtually zero, but in Europe, their policies are similar. So I think they, in some ways, look to human experience. So, for example, if you have a phase 0 clinical trial happening in the United States, the European regulators tend to, see that data. For some things, actually, they are much faster. But I think in general, it's not that different. I think people realize that this is an important problem, and they tend to not stand in the way. But the problem is, a lot of these things are expensive. I mean, taking a new therapeutic into humans could be, for many companies, is like a multi-billion-dollar process. Many of the drugs they develop never make it. Probably 99% of them don't even make it to a clinical trial. I mean, many of the drugs that are made fail like on the way. There's this kind of valley of death. And I think if you're outside of a big company, like a startup, for example, getting into that clinical trial, that's like a huge milestone. That's why a lot of these startups get bought out by big companies. Some of them even before they start their phase 0, because just the idea that they made it all the way to that is a huge advance for a drug development pipeline for a company. So, for those of you who are interested in, entrepreneurial things, having a good idea and getting it to close, even like through safety talks, like close to a clinical trial is still like a big milestone, could be an exit for many people. So something to think of. Many companies have gone public that didn't have anything, like nothing, and they made tons of money. So there is a huge push, and once there's money in something, these clinical trials happen. The FDA will not stand in the way of something which is good. So that's been my experience with INDs and trying to get the FDA on board to help do things. But usually it's the bigger problem is money, like, at least for me, but not the FDA. Like, you need a lot of money to do a lot of these studies, and usually that requires partnership with industry to make that happen.
Thanks, Kevin John. Glad to have you here. I do appreciate if you share your thought about clinical challenges or path to clinical transition of hyperpolarized MRI, especially in children and pregnant women, and also what is your perspective for near future. Thank you so much.
Sure. In terms of children, so for maybe one kind of the idea of translation. So I think for the clinician, the idea would be, do we have a molecule which is safe and tells you something that is interesting? So I think this first molecule that we translate to humans, pyruvate, has some interesting characteristics and is something that many clinicians are interested in, and they're trying to translate, and there are a lot of ongoing clinical trials. So in terms of pushing that forward in kind of adults, I think that will happen pretty seamlessly because it's kind of already happening. I think there the idea is how well does the field expand in these clinical sites beyond traditional academic medical centers, kind of these university hospital type places, to the broader reach. So I think that's the challenge for that field. But in terms of kids and pregnant women, so it's very exciting that there's a number of studies that are ongoing, a few that have actually come out. The imaging of DIPG patients, so kids with deep intrapontineal gliomas, they're brain tumors in basically the middle of their brains. And so these kids tend to be, under 10, so say like the 5-6-year-old to, I think at the oldest, maybe you'll see them in the low teens, 11, 12 years old kind of range. And so there have been a couple studies already in these kids, which, have shown kind of, as far as we know, no kind of adverse toxicity. And this kind of makes sense because the substrates that we inject are endogenous. They're normal appearing in your metabolites in your plasma. So we're not injecting a weird thing. They so far tend to be safe at the doses that we give. So I think for kids, the problem is more actually not related to the technique. It's more about imaging in general. kids tend to move. And so, below a certain age, imaging kids is very hard. And typically, they can be put under anesthesia because we need them to not move around while you're trying to take a picture. I mean, if you have kids, I have a 3-year-old, getting her to stand in one place so that we can take a picture is already very hard. Imagine trying to put her in an MRI machine and trying to get her to not move around. So I think that's the challenge. There was the challenge of CT and MRI already. In terms of pregnant women, we also think that it will be safe for them. And so, the same way that MRI is safe for pregnant women, like 1.5 Ts are routinely done on pregnant women, we think that, unlike radioactivity, which I think would have a problem, maybe, these molecules are safe, we believe, stable, and have no, they're cleared from your system. Actually, pyruvate has a half-life in your plasma of about 15 minutes. So in an hour, it's mostly gone. And this is not even about like our imaging component. So I think that it's actually very likely to be used in pregnant women. And there are even preclinical studies of pregnant rats or imaging the fetuses of rats using this technique. And so I think there's a lot of interest in that. You could imagine that trying to understand baby metabolism in situ, like in a pregnant woman, would be really important for trying to understand development. We do very rudimentary things actually with ultrasound that are very important when kids are developing, measuring bones, measuring, aside from how many fingers and toes they have, but, function of the baby brain or function of the baby heart could be things that we could observe in a pregnant woman. So I think there is a big avenue there, but most of the applications right now are much less, much lower risk. So, people with cancer, the pyruvate is not their problem. They have cancer. They have, unfortunately, bigger problems. And I think adults generally, it's lower risk than going for kids or pregnant. But I do think that is a direction that definitely will happen.
خیلی ممنون. یه سوال خیلی جزئی‌تر در مورد همین روش HPMRI که استفاده می‌کنین، در مورد ریپرودوسیبیلیتی می‌خواستم سوال بپرسم. تو همین در واقع تحقیقتون آیا چالشی داشتید با بحث ریپرودوسیبیلیتی برای این فناوری؟ اگر آره، تو چه زمینه‌هایی بود؟
I think almost all imaging generally has this problem of reproducibility. I think this is what's made things like machine learning so hard, because the first thing you need to do is some level of normalization to be able to normalize the data. So that we have, I think, in this kind of new evolving field, multiple parts of this reproducibility problem. One is that, just robustness and reproducibility of the scanning itself. But because our imaging is single shot, that we don't single average, it's challenging to repeat the same exam. I mean, we can re-inject the same person and image them again, which we have done. And it turns out the variation between these scans is not very much. Meaning, like, if I inject somebody, image them. So we did this in this cohort of prostate patients where we imaged them, and then they were laying on the table. We imaged them again, like an hour later. So I said, the half-life of pyruvate is about 15 minutes. So we were like at 4 half-lives, and the FDA said that, oh, that's okay, you can inject them again. So we did that. And we found that the variation between the images from these injections was less than 10%. So the reproducibility in a site, doing it the same way, the variation will probably be similar to what it is for other tracer type studies. So, in the classic kind of example is in the setting of things like FDG PET, where, many people are injected with FDG and imaged over, large cohorts, and the variation in FDG for retest reproducibility is about the same, 10 to 15%. I think, that takes into account, dosing differences, slight differences in measurements, slight differences in equipment, so on and so forth. I think for us, because the parameters that we would quantify are not well described, it's unclear what imaging parameter, because we can measure different things about the image. We can measure we can quantify different parameters, how fast molecules reach a target, how much they get converted, to what rate, and what properties of these kinetics you try to quantify, they all have some susceptibility to, reproducible error. So, determining which parameters are the most robust invariant to all these different problems, I think, is something that many, many clinical trials are going to do, and lots of studies will try to figure out. So we have that, and then, of course, with such a small number of labs and sites doing this right now, everybody kind of does it a little bit their own way, and I think that eventually the field needs okay, this is how everyone does it, and let's make it standardized. And I think kind of like a NIST standard that we will do it this way, and you will quantify this parameter this way. So I think once we get to that, then I think the robustness, the reproducibility will get better. This in the pet field, this happened, and they developed parameters that were measured a certain way. So, for example, SUV, or standard uptake value, is the standard measurement that's made for FDG. So on a lesion, they measure the maximum SUV, they might measure the mean, they might measure the median, but SUV becomes like the basis for, reproducibility and normalization. And I think our field needs a similar thing, and that's controversial, but, I think we will arrive at that parameter. We just need to measure enough and do enough people to figure out what that parameter is. And it might be different for different tumors, and it probably will be different for different molecules. So, because different molecules turn into different products, and they can have cascades. So I think we're so new that we don't know that yet. So I think maybe you don't want things to be so defined at the beginning, because you will lose out on, contrast that maybe we wouldn't, phenomena that we wouldn't know about. So right now it's open, but it does make a challenge to reproduce exactly what other people do.
خیلی ممنون. بله، این بحث همیشه مطرح میشه تو اکثر فیلدها. گروه ما هم که با در واقع ریسرچرهای مختلف اینجا کار میکنه، همیشه رو بحث حالا مهندسین افزار پژوهشی این مشکل ریپرودوسیبیلیتی هست. حالا حتی برای فرض کنید فیلدهای مثل پابلیک هلت که دیتای روباست هست، اما اون آنالیزی که رو دیتا انجام میدن توسط محققین، خود اون ریپرودوسیبیلیتی، اون پایپلاینی که اون آنالیز رو انجام میده هم مشکلات ها برای شما، که خود اون بحث دیتا مشکل خواهد بود. بحث ریپرودوسیبیلیتی یک کتابی رو شما در 2018 به چاپ رسوندید، ویرایش اول، در مورد تصویربرداری و متابولیسم، ایمیجینگ و متابولیسم. ممنون میشم که در مورد این کتاب کم توضیح بدین. آیا این تکست بوک هست؟ این رو به چه کسایی در واقع توصیه میکنید برای خوندن؟
Sure. So that book I co-edited with my friend and collaborator, Jason Lewis, and he is a pet radiochemist, and obviously I'm more MR focused, and so we decided to write that book to really introduce the kind of idea of imaging metabolism as a framework for many different disciplines. So you could approach that, I think, as not as much of a textbook that you would take in a, let's say a class with problem sets, but more so a textbook, a book that can introduce you to metabolism and imaging multimodality across many, many different diseases. So I think it could be, and maybe closer to the question that I think someone asked before about, resources for trying to understand things like GBM. This is probably a nice resource for trying to be introduced to many, many different approaches and techniques for imaging metabolism, a lot of which is in vivo in humans, which has been translated to humans, but also that can be used preclinically. And we'll give a kind of a primer also. There's a great chapter towards the beginning of the book on cancer metabolism, to give, at the time that book was published, kind of what the state of the field was in terms of metabolic processes in cancer, but also primers for other diseases, and then imaging techniques that were matched to those. And I think you can, if I remember correctly, you can actually also just download chapters of the book. So there is Springer, which is the publisher of that book, they had started a new thing called Springer Connect, I think, at the time. So there's if you're a part of a university, or the university library system, they have a they have an agreement with Springer, and you can actually just download chapters or the whole book as a PDF, and you can just read all of it. I would encourage you to just check it out. Maybe some of what's in there, especially references that those chapters will highlight, they may be useful, for really kind of bolstering and understanding of some of these things. And if you're interested in a lot of these different directions, you might find some of those chapters very helpful. I think especially I think, if there's biochemistry, if you're an engineering type of person, the biochemistry may be challenging. That cancer metabolism chapter is actually written in such a way that it's both accessible and very informative. I think if you're a biochemistry person, you're wondering about, how do these techniques work? How does PET work? How does hyperpolarized MR work? How does SPECT or optical imaging work? There are actually great chapters, I think, in that book that describe kind of the strengths and weaknesses and how those processes work. So it is a useful, I think, set of kind of data and explanations for a lot of things that are relevant to these kinds of questions that my lab and many other labs are interested in.
بسیار عالی، خیلی ممنون. شما اشاره کردید که لیسانستون رو اینجا بودین برکلی. آیا تجربه تحصیل هم در ایران داشتید قبل از این، یعنی که از ابتدا های اسکول و یا قبلش رو هم همینجا بود؟
نه، من همش رو اینجا کردم. I grew up here, so I never actually went to school in Iran. So I don't have experience with that, though many people in my family have. I think, and I've had students in the lab who also came from Iran. I think there are similarities. I think the push here for us happens later. I would say that most students in Iran are challenged much more earlier in your training, like in middle school, high school, but maybe to a higher level. I think the bar, especially for many of the schools that, kids graduate from high school in Iran and come here, they tend to be a little bit more advanced than the average high school student. But I think that gets made up in college. But I think like in the first couple years of college, everything becomes kind of the same. So that's been my experience with high school and college. Though, I mean, there are many of these high school programs now that are like college in the U.S. So I think, they're definitely both very strong. I think in both situations.
بسیار عالی، مرسی از توضیحاتتون. حالا میدونم البته مطمئن نیستم کدوم در واقع شما توی شعبه نیویورک هستید، درسته؟ به همین مموریال.
Yes, I live in Sloan is in the Upper East Side of Manhattan, so we are a couple blocks from Central Park.
بسیار عالی.
And I live actually in Manhattan as well. I walk to work. It's great. It's my favorite thing.
خیلی هم عالی. منم چند سالی تو منهتن بودم. فضای منهتن یک برخلاف حالا جاهایی که خیلی آکادمیک هستش، بیشتر دامیننت هست با این فاینانشال اینستیتوت ها. یادم سال 2011 یک کاری بودش توسط شهردار منهتن که یک کمپس تکنولوژی بیان توی منهتن در واقع بذارن و کمک $100 میلیون میکردن که در انتها کورنل بنده شد با در واقع تکنیون اسرائیل اینستیتوت آف تکنولوژی. حالا برای شما تجربهش به چه صورت هستش؟ مطمئنا همکاری‌های در واقع ریموت که خب با ابزارهایی که هستش اشاره کردید شما راحته، اما خب دسترسی و مشارکت حضوری هم خب میتونه خیلی تاثیرگذار باشه. مثلا تجربه شما تو زندگی و تحقیق در منهتن برامون بگید.
So that campus ساخته شد رو چیز there's an island next to us called the Roosevelt Island, and they built that campus on Roosevelt Island. So we call it the gondola. It's like the thing you take to go skiing. There's this little telecabin thing that goes from right on 61st Street, and it goes to Roosevelt Island and drops you off at that new campus on the island. Actually, I think it's like right there. So I have actually never been there, even though we live very close. Most of the people that ended up being on that campus are, I think, data analytics, computer science, statistics type of people that were originally at the Ithaca campus. We don't have very much interaction with them, to be honest. But I think that it's just recent that they've started to build that up. In terms of the rest of Manhattan, though, I think we are very integrated, and it's a great place to do, I think, to live and to do science. I mean, obviously, we have this problem of it's very expensive to live in Manhattan, and even the outer boroughs. But I think that it's actually not as bad as some other places, like Palo Alto and San Francisco, for example, are extremely expensive and they have no amenities. So I think New York has all the amenities of a big city and the connection to the rest of the world that I think some of these other places don't have. But we also have a very strong collaboration with all of these other universities because we're so close. So, I mean, Rockefeller University and Cornell University and us were all like literally on the same block. But then, and we would say that's, the furthest place maybe is like NYU, which is just, I don't know, we are on 68th Street, 67th Street, they're on like 40th Street. So it's about 20 blocks away. I mean, if you really want to, you take the subway there in 5 minutes walk. I mean, Columbia University is just on the other side of the park. It's also not that far away. So, I mean, we have this, as well as like many other smaller, there's the City University of New York, and they have a number of campuses. There's one actually very close to us called Hunter College, which is very good. So I think the university structure in just the island of Manhattan is huge. It's like very dense. And so I think that creates huge amount of possibilities. We recently started collaborating with a lab at the Advanced Science Center across town, which is part of the City University of New York, where they have a very strong program in, implanted, what are called optic color center or NV centers and diamonds and other materials work. And so, there are these kinds of facilities all around the city that are very accessible. I mean, in most places, you would only have some of this in one university, and everything else would be very far away. And so, the science aspect of here is like very dense, and I think also all the cultural aspects of Manhattan. So it's actually a great place to live. You have to get a little used to the kind of apartment lifestyle. If you're not used to apartments, then maybe Manhattan is not for you. But outside of that, I think it's actually a very powerful place, and it's very exciting. Kind of vibrant. Everything is open all the time, even during COVID. Let's say after, well, I don't know, post the apocalyptic COVID thing, you can walk around. Still lots of people. Everything is open. Things are happening. So I mean, this is a different. You don't feel this energy anywhere else. Like any other. I've been all over the world, and I don't think I see this kind of energy anywhere like I do in New York City. So it feeds into everything.
عالی. مرسی. بله، دانشگاه دیگه هستن تو منطقه‌ای که میشه باشون کلابوریت هم کرد. مرسی از شما. حدود 2 ساعت هست که مهمون کلاب ما هستید و خیلی استفاده کردیم از صحبتایی که انجام شد و بخشی که دوستان فارسی حرف زدن و حالا خوشحال بودیم که ترکیب فارسی و انگلیسی خیلی جالب شد. اگر به عنوان حرفای پایانی واسه کسایی که دوست دارن در مورد فیلد شما کار کنن توصیه دارین یا حرف پایانی، خوشحال میشیم که
Yeah, again, if anyone is interested in any of the things we talked about, please feel free to email me. I'm happy to talk about anything. If you're interested in the lab or you're interested in science, please feel free to shoot me an email or follow the lab on Twitter. We have a Twitter account, which is I think just at Keshari Lab, and I'm just excited to meet everyone, and thank you so much for the invitation to speak with you. And I'm glad I could give it a little bit of a different spin. If everybody comes and speaks Farsi, then it will all be the same. So now I forced you to to also use a little bit of English. It's helpful.
مرسی، خیلی ممنون از شما. ما هم خیلی از شما متشکریم که امروز به ما جوین شدید و ملحق شدید و تحقیقات و تکنیک‌های نوآورانه خودتون رو با ما به اشتراک گذاشتید. من دوست داشتم همینجا از دوستان حاضر در اتاقم تشکر کنم که تا این لحظه با ما بودن و هر جای این دنیا هستین روز یا شب خوبی رو برای همتون آرزو میکنم.
خیلی متشکرم کیوان جان بابت صحبت خیلی خوبتون. من خیلی لذت بردم. متشکرم.
منم به نوبه خودم تشکر میکنم. خوشحالم که امروز باتون وقت بیشتری گذروندیم و آشنا شدیم با کارات.
مرسی. منم همینطور روز خوبی داشته باشین. حالا انشالله به زودی همتون رو میبینیم in real life. Maybe if you come to New York, feel free to look us up.
Absolutely.
بسیار عالی، مرسی از شما. اتاق رو 30 ثانیه باز میذاریم تا اگر دوستان میخوان که کلاب رو فالو کنن یا اینکه با دوستان مادریتور در تماس باشن یا با مهمون برنامه، و بعد اتاق رو میبندیم. باز هم تشکر میکنم از کیوان عزیز که وقتشون رو در اختیار کلاب گذاشتن. اتاقهای آینده کلاب هم به محض اینکه نهایی شدن در واقع اسکیجول میشن و شما میتونید در صفحه کلاب از این اتاقها با خبر بشین. روز و شبتون بخیر هر کجای دنیا که